anatomy to continue your anatomical journey through the day. What's the body called on this sucker? Point it out here. This whole thing is called a what? Strobila. Yeah. And since it's a segmented worm, what are the individual segments called? Troglodids, yes. And then this head-like structure that doesn't function as a brain is called a what? Stolix. And it might have suckers on it. 4 different suckers. Alright. Oh wow, this is really small font. You guys can't read that at all, can you? Alright, well that's not very fair if you can't read it. We'll just pretend like I'm asking you and you can't see that on the screen. What is the stool exam used to look for parasites called? OMP, and that stands for, what's the OMP stand for? OPA and parasites. We'll talk more about that when we get to the blood deck. Okay, yesterday when we started talking about the tapeworm specifically, the first one we talked about was this one. It was Taenia solium. What's the common name for Taenia solium? Cork tapeworm, yes. There were 2 different forms we talked about. This question is asking about the intestinal teniasis form. In that form, what's the definitive host? Humans. Humans. How do you know it's a human? Yeah, so for basic definitions for parasitology, the definitive host is the one where sexual reproduction and maturation occurs. So if you think about that life cycle diagram where that person is eating the infectious source, which we'll get to in a question or so, that maturation of the adult worm occurs in a human. So that makes the human the definitive host for intestinal teniasis. And then the intermediate host would be which animal? The swine, the pig. Yes, good. Oh, they're out of order. So what's the infective stage of that parasite that causes cysticercosis or neurocysticercosis? Is it the eggs or is it the larvae? It's the eggs. It's the eggs that causes is generally considered to be more serious but rarer form of cysticercosis. And then this is a lot of word, but the infective stage of Taenia solium that causes intestinal teniasis would be the larvae. And what's the word for the larvae when we're talking about these cestodes? Like pork tapeworms, 6 weeks? It's close, but we're getting warm. Remember, it is the Cysticercus, plural side. So that's the larval form that's infected, just a normal thing. And you find that in what kind of source? Undercooked or uncooked pork. When you have this intestinal form that causes usually asymptomatic mild diarrheal symptoms, what stage of that particular parasite is diagnostically useful for somebody doing an OMP prep? Proglodids would be useful. Yeah, we can stay in that. We can count the number of uterine branches. What about eggs? I mean, maybe a little bit, but what can't you differentiate from? It looks the same as the other tapeworm we talked about that's similar, right? The Taenia. The beef tapeworm is saginata. So you might be able to say that's a Taenia species, but you can't tell which if it's solium or saginata. Those eggs. Proglodids you absolutely can. How is it diagnosed? We just went over it. How is it prevented, that intestinal form? Yeah, cook what you eat. Don't do this horrible, raw bacon thing or whatever. What about preventing cysticercosis form? Good hygiene. I'm sure you don't transmit that eggs, so if you get the eggs on your hand and you don't wash your hands after you go to the bathroom, they can end up in food sources and water, which is just typical fecal oral route transmission. For the diagnosis of that cysticercosis form, how would you do that if a patient had the cysticerci encysted in their muscle tissue? Imaging studies, anything else that you might be able to do? What about a biopsy? Do a muscle biopsy and look to see if you can see parasitic structures there? Yeah, and then serology too, perhaps. Some value. All right. Common name for Diphyllobothrium latum. What is that one? The fish worm. Yes. What's the definitive host for that? Think of the signs when humans get infected. Signs and symptoms. What are the signs and symptoms if somebody has diphyllobothriasis? Yeah. Yeah, and so some ways to dental crumping. Does that sound like an intermediate type of thing, or does it sound like a definitive host for that tapeworm is maturing to adulthood? The latter. So we are the definitive host for that. We talked about the symptoms with Diphyllobothrium. What else is kind of unique with that particular infection, that tapeworm? The vitamin B12 deficiency, yeah, which is more pronounced with that infection. The eggs, there is something unique about the eggs that come out of us when we're infected with Diphyllobothrium. You guys remember anything distinctive about that? They're percolated, which is a little unusual for sustenance. Not unusual for trematodids, but for sustenance it is. Spirometra species. Mancinoides. What's the common name for those? Snake tapeworms, and the definitive host for the snake tapeworm? Hosts. Cats and dogs. Yeah, cats and dogs are the ones who get that adult tapeworm in them. It's really we're accidental hosts and we're dead-end hosts. Since we're not the definitive hosts, how does that present in a human if we come across it, if we get that snake infection, sparginosis? Skin nodules all over the place, depending on how you acquire it. What if you were to apply a poultice to your eye? You can get those larvae migrating into your eye and potentially all the way through the brain. Yeah, but the point is, you're not the definitive host. It's not going to cause intestinal issues. It's going to flounder around under your skin for a while until it dies. Echinococcus granulosus, common name for Echinococcus granulosus. Dog tapeworm, and the definitive host for this one? It's the dog. Intermediate host? Sheep? Yep, good. And we are accidental hosts and intermediate hosts. So we develop these these larvae that grow in these cysts, hydatid cysts. You get to be really large, just like a fruit sometimes. How might you diagnose somebody who has the risk exposure for echinococcosis? Like if somebody has something the size of a grapefruit slowly growing inside of their liver or some other important organ, what would be a good way to find out what that structure is? Imaging. Yeah, you do some imaging, MRIs, CT scans, ultrasound especially, with echinococcosis. Okay, Echinococcus multilocularis. What is the common name for that? Fox tapeworm, yes, and the definitive mice, rodents, rats, yep, good, things that they prey upon. When you want to diagnose a VLR, echinococcus caused by Echinococcus multilocularis, how would you diagnose that? It's not a bad guess. Imaging, perhaps some serology. What are you, with multilocularis, that the name multilocularis, multiple locations, these VLR cysts, and added cysts can go into multiple locations. And as we borrow a term from the field of oncology, metastasis, use it for this particular parasite. Parasite has a more aggressive infiltrative nature to it. And we worry about it infiltrating into the tissues. So pathologists sometimes mistake it for cancerous, but it's technically a parasite. Hymenopus nana, common name for that? Dwarf, yeah, the little one, the dwarf tapeworm. Our humans different hosts. It's kind of a trick question. Not required. So the invertebrate arthropod intermediate host required are optional. They're used to kind of amplify those portions of parasites in our brain stores and contribute to the problem. It's one of the most common parasitic infections, but they're optional. So humans are the definitive host for this. They grow to adulthood in humans. And then Hymenopus diminuta, that was a common name of that tapeworm was what? Rat tapeworm. What was the intermediate host for the rat? The flea. Very good. Yes. Is that optional or not? Not optional. Flea, not optional. Must have flea. Common name for Diploidium caninum. So the last rat tapeworm is Hymenopus diminuta, but Diploidium caninum is commonly known as the flea tapeworm. Very good. Okay. And the flea is what? Definitive or intermediate? Intermediate. Yeah. Okay. What's the definitive host for the flea tapeworm for Diploidium caninum? Dogs and cats, exactly. So humans are what kind of hosts? Dental hosts, correct. Yeah. How would that present in a human? Actually, we can transmit. I'm sorry. Yeah, we can transmit. How does that present if you have the Diploidium caninum in a human? Diarrheal symptoms. You have the tapeworm growing in your intestines. Okay, and then if you have this infection, Diploidium caninum, and you pass eggs in your feces, what is diagnostically distinctive about those eggs? Yeah, it looks a little distinctive. Okay, and then probably the most important question on this review slide, which two species are at risk of auto-infection? Meaning that you can be the source of your own infection. Getting even worse infection, hyperinfection. Teniasolium, yeah, you can actually auto-infect Teniasolium. And then, yeah, Hymenopus nana, yeah, the one that has the optional beetle and rat, or rodent intermediate hosts. Okay, so that's a lot from yesterday. Just kind of studying together things you might want to have a good grasp on. Trematodes. These are the flukes. We have a couple of them to cover today, going over a couple of basic features of what trematodes are. Most of these trematodes will have a very flat, sometimes described as fleshy, as to the leaf shape, a lot of while you're studying flukes. Remember, flukes, flat, fleshy, leaf shaped. They do have two suckers on them. One of them is an oral sucker, and they do use that as a mouth. So in terms of their digestive tract, it's a little bit more evolved than the digestive tract of cestodes. Cestodes obtain nutrients through like what means do cestodes require nutrients? Absorption through their skin or their tegument. With these guys, they have a mouth, but they don't have any excretory opening. Yeah, so that means, again, like the timing when they eat, very important. There's only like one way in and one way out for waste. They also need to have a ventral sucker. See, we have different types of flukes that like to adhere to different parts of the body, but they need to be able to latch onto those different organs and as they develop. Most of these guys are hermaphroditic, but meaning that they can self-fertilize, they can have both female and male components. But in the latter part of this deck, we'll talk about schistosomes, which are unusual for a number of reasons. One of those reasons is that they do have separate genders. They have males and females. We call that dioecious, separate genders. And their body types are unusual too for flukes. They don't look like the rest of the flukes. All of these guys require snails as intermediate hosts. And we're not talking about terrestrial snails, we're talking about aquatic snails. So for these infections to occur, you have to be near bodies of water where snail populations are. That's a common thread with these. Another common thread is most of the eggs are percolated, with the exception being the schistosomes. Here again, there's an percolated egg. You can see that lid on the top of it. Very, very distinctive. And the rest we're going to talk through some of these life cycles. This is a nice overview slide of the intermediate host, snails and all of these guys. Some of them only require a snail, but some of these infections, like Clonorchis sinensis, will require a second intermediate host, like a fish, or Paragonus westermanii, that has crabs and crayfish. Just a little bit higher up the food chain, they have to eat those larvae that are released from snails in order for it to be infected for humans. Let's talk about that infective stage. In terms of target organs, we've got the Phaseolopsis buski, which is the only one on this list that targets the intestines. And a lot of the symptoms when we talk about this one are going to be specific for intestinal issues. We've got two liver flukes, the Chinese liver fluke, and then we've got Phaseola hepatica, the sheep liver fluke. So those symptoms will be somewhat reminiscent of damage to the stems of the liver, hepatitis, jaundice, things like that, liver problems. We've got a really unusual lung fluke, so we get some respiratory involved there. And then lastly, schistosomes, really, really important. By far the most clinically important member on this list would be schistosomes in terms of morbidity on the human population. This is a blood fluke. It gets its nutrients from being in the blood vessels around either the liver or the bladder, depending on the specific species of schistosome that you're talking about. Biological vectors is good for us to study, but kind of when you think about the biological vector, this is what is important for us in terms of the source of these larvae. When we talk about the first couple ones, Phaseolopsis buski and Phaseola hepatica, these insist on aquatic plants, right? So that's the biological vector. It just means that this is how we're acquiring that larvae most of the time in these infections. And then the reservoir hosts, the more hosts that are involved in sustaining these trematodes, the harder it is for us to eradicate this from the human population, generally speaking. Alright, so we'll start off with Phaseolopsis buski first. Causes Phaseolopsiasis, simple word, big word. This is arguably the least clinically relevant disease on this list of trematodes, but it's not trivial if you're living in one of these endemic areas in Southeast Asia. You don't get these cases in the United States, but it is important in Southeast Asia, and it's important because of the agricultural practices that people have there. This is associated with snail species that live in Southeast Asia, and those snail species become effective once they're Cercaria. They go on, they're out in the middle of the water or whatever lake. They go out to some aquatic vegetation on the outside of the pond, where they know that if they insist on these water vegetation sources, they're going to get eaten by some animal. So some of these animals, pigs, dogs, rabbits, whatever is feeding off of that water vegetation, can ingest those insistent larvae on chestnuts are oftentimes implicated as a common source, but it can be other types of water vegetation as well. Water chestnuts, if you've never had them before, they're really, really healthy, high water content, very fibrous, less seed, super healthy, but when people are eating directly out of the water, A, it's really bad for your teeth, but they're peeling the husk away and eating the meaty part of that water chestnut. And B, that's an enriched source for these cystic larvae. So you eat the larvae on these plants. Larvae will exist when they come out of that protective cyst that they're in. Once it gets the upper part of the small intestines, and then they'll attach to the small intestines where they'll start maturing to adults. This time, from the time that they attach to the small intestine to the time that they are mature and start self-fertilizing and producing eggs, the diagnostic value takes a couple months. So if you have expected exposure, it's going to be a while before you see anything in the O&P exam from those patients. The cycle continues when humans dump their waste into areas that can be, that can trickle and flow into bodies of water. But even if you were to stop transmission from humans, you have these reservoir species that contribute to this transmission cycle, so you can't really stop it by breaking that transmission chain for humans. Once these get out to the water, those Neuracidium have evolved to be able to penetrate snail species. So they'll get inside the snails, they'll do terrible things to the snails, if you care about the snails. And then when they've developed, they'll come out of the snails and be in this form called Cercaria. So they emerge as Cercaria, they come out, develop further in the water. There's these tail area that have tails that allow them to swim to the shores of those lakes. And then they lose their tails once they insist on these water chestnuts, whatever vegetation. In terms of symptoms, if you have a light burden, which means you have ingested a few of these larvae, you might have asymptomatic infection. If you're ingesting a lot or frequently ingesting a lot of these larvae, some of these more severe infections, including diarrhea, abdominal pain, they would be associated with the development of these adults in these intestines. Let me just show you this really quick. This is a magnified picture of what you have in the upper right corner of your screen. These are these giant intestinal flukes. You don't have the blown up one of the paper next to it, but they're giant. They're called that for a reason. They're big. You can see them on your hand if you were to hold them up. These are big flukes when they're adults, so they do some damage to you. This is a heavy infection in the colon. You can see when the intestines are there. You can see a bunch of these adults on there. And they're not just eating your food, right? It's not like they're just siphoning off calories like tapeworms. They're also feeding off of your mucosa. So there's some damage. There's more significant damage done to you than these trematode infections generally speaking. So these symptoms are a little bit more severe, and they can start interfering with your digestive tract ability to absorb. Some of those symptoms that can be as bad as giardiasis and malabsorption disorders. You can no longer digest food, basically. When this happens, adults can stay in there for about a year, so you can imagine having a heavy burden for about a year. Some of those symptoms can be pretty severe. Very, very heavy case, likely a little bit of obstruction in the intestines. And you might get some systemic symptoms as well when the body's immune response tries to clear these flukes out of your intestines. Let's see. Abscess at attachment sites, because it is wounding you, that sucker, that medical sucker is causing a little bit of injury. Ascites, I don't know if we've talked about ascites before. What is that? In the cavity, in the abdominal cavity in this case. So that's when patients who have ascites start having like a distension in their midsection where fluid is collecting in their abdominal cavity. Going back to the systemic symptoms, because these worms can get to be heavy worm burden, they're also releasing a lot of waste. So that waste is toxic to you. Once you have a significant amount of that waste being released into those will contribute to the systemic symptoms migrate to the liver, as the name suggests. So the symptoms are going to be more reminiscent of liver damage. It's nicknamed the sheep liver fluke because sheep are a major reservoir for this particular parasite. Sheep will graze on aquatic plants that humans like to in certain parts of the world. So the prevalence of fascioliasis is bigger in sheep farming parts of the world. Different sheep populations. But kind of the same story. Instead of a water chestnut, the water cress is an aquatic plant with Fasciola hepatica. You consume those, they develop in your liver, and as they are developing, the adults develop in your liver and bile ducts. You get symptoms that are indicative of that liver damage. These worms, the immature larvae, will migrate through your liver. During that acute phase, you'll get symptoms like some right upper quadrant pain of your liver. Literally has larvae migrating through it and doing damage to it. Some hepatomegaly, fever, jaundice. Let's see. They can be there for a while, and you can enter this chronic phase, especially if bile ducts are affected. In the chronic phase, you still have that right upper quadrant pain. Obstructive jaundice, if your bile ducts are obstructed, it means your bile is not released, and you get cholangitis, cholestasis, the lack of flow of bile, fibrosis. You get secondary infections, chronic parts of your liver that contributes to what we call liver rot, which can be really serious as well. So you start to lose liver function, portal cirrhosis, very, very serious complications like that. Secondary bacterial infections because the liver is so torn up from having infections like this. Once the adults reach maturity, they will start producing eggs. For the eggs, they are virtually indistinguishable from the eggs of the last species. So Fasciolopsis busky, the intestinal fluke, has very, very similar eggs. So that would be a complicated diagnosis. You might have to look a little bit deeper than that, maybe do some serology so you can get a definitive diagnosis. I think the treatments might differ because the intestinal fluke could probably be cleared up across Quantal, but I don't know if that would be an appropriate treatment for Fasciola hepatica. Okay, Clenorchis sinensis is another liver fluke. This one has a secondary intermediate host of freshwater fish. Pretty significant burden worldwide, 15 million infections, mostly in Asia. Anything implies Chinese liver. We have a lot of other hosts that like to eat raw fish, like cats and dogs, so there are lots of other reservoir species besides humans. But enough humans eat undercooked and uncooked fish in those regions where we get a significant amount of caseloads worldwide. So we eat these fish. The fish have larvae in them. The larvae will exist in our duodenum. Once they exist, they latch on to our bile ducts, and like small intestines, make their way into the bile ducts and grow to adulthood. This teeny tiny ruler shows the size of these adults. They're also fairly good sized flukes. They'll migrate into the bile ducts and start producing eggs at that point. So the eggs themselves that Clenorchis sinensis produces are super good. They're large for one thing. They also have a pronounced perculated lid. And then on the other side, you guys see that little knob on the other surface? That is called an abelpercular knob, and it's distinctive for Clenorchis sinensis. So if you can see that, usually you have no problem diagnosing this infection. Once you release those into the water, instead of actively burrowing into susceptible snail species, these are actually eaten or ingested by the snail, a small minor detail, but otherwise that infection cycle is pretty much the same. Question for you guys about transmission. Say you were to, for whatever reason, eat a snail that was infected with this larval stage, do you think you would become infected? Why or why not? This secondary host maturation, and that further maturation is contingent upon those specific cues and signals it gets once it's ingested by a fish, basically. Okay, so if you love astrophotography and you're freaked out about getting some sort of trematode, don't be. That is not how that works. They require further development, and that includes species that don't have an intermediate host, because even those have to become cercaria, free swimming cercaria, out in the water. Okay, in terms of symptoms, the symptomatic presentation, like we're mildly symptomatic, when you get heavier infections, you get a lot of fish, there's larvae in them. Most of this reflects the liver damage that you would see as these adults start maturing and producing eggs. You also get mechanical irritation from the words feeding off of your liver, so that's where you're getting a lot of nutrients. They're latching onto it, causing irritation, migrating all over the parenchyma of the liver, releasing toxic byproducts that can cause systemic effects of fever, for example. They can also wound as they're living and eating your liver and your bile ducts. This is causing a loop of wound healing repair cycles, and that can contribute over time with chronic infections to cholangiocarcinoma, a bile duct cancer. This is the first time we've encountered a parasite that can contribute to a carcinoma, but since chronic infections can do that. You can also get gallbladder involvement, cholecystitis, cholelithiasis, gallstone formation, other impairments of functions, liver abscesses that make you more vulnerable to secondary bacterial infections. There we go. Over a centimeter for the adults. All right. This organism needs a second intermediate host as well. The second intermediate host for Paragonus, and they're usually like crustaceans, crabs and crawfish specifically. We do see cases of this in the Americas, occasionally in the United States as well. We have parts in the South that are fond, particularly eating raw crayfish, crawdads, things like that. So it happens occasionally, but very rare in the U.S. Much more common to see Paragonus in Asia and Africa. So here is, like I said, it's a lung fluke, so this has a pretty involved migration that it has to undergo. You swallow it, it migrates out, for instance, it goes into the liver for a while. These are teeny tiny larvae, right? So they're small, they haven't matured into adults. Once they get out to the liver, they get picked up into circulation, they move through the diaphragm, up through the abdominal cavity. So, and this is kind of confusing on this figure, but after they get through the diaphragm, they briefly go to the heart and end up in the abdominal cavity where your lungs are. There they land in your alveoli, and you get signs and symptoms associated with respiratory damage at that time. So, very convoluted infectious cycle, but the lungs is where they take up residence long term. I think I have it on here how long their lifespan lasts in the lungs. It's a considerable amount of time. So early on during this migration, fever, chills, high eosinophilia associated with this. Once it gets to your lungs, you'll be coughing. Increased sputum. If you were coughing up these adults, see that in the sputum, so the diagnostic specimen is a value of the sputum itself, or if you swallow that back down, then you're released into the stool. So you can use either sputum or stool to look for this parasite. Their ability to go into these ectopic sites like the brain, spinal cord, sometimes other organs, is fairly rare, so I won't talk about the presentation of that, but focus mostly on what can happen with presentation of the lungs. They can also, as they're migrating to the lungs, they can cause bleeding in the bronchioles, so that's where you get this characteristic blood-tinged sputum, because you're bleeding a little bit and that's getting into your sputum. Do you have a question, Justin?
I was curious, like how
I think that the presentation itself is fairly different. Or if you suspect a TB, you can rule out TB at a bunch of lab tests for rule outs of TB. Notes in terms of those sources of infection, crabs and crayfish, those raw, like meaty crustaceans that you might eat. Some parts of the world they come over, they'll soak their crabs, crayfish in wine, but that again is done as a preservative. It limits the bacterial growth in those food sources, so it does nothing for parasites. I mentioned that yesterday, but keep that in mind that parasites don't seem to mind being pickled. Yeah. Yeah, I don't think they're killed. This is like being coughed up is a normal thing for them. So if you swallow them back down, they should be perfectly viable through that entire process. They've adapted pretty well. They're good ones. Before I'm going to put you guys on break in a second, I do have a couple of videos before I put you on break. And this is after lunch, so you should be totally fine. Hopefully, okay, so what is it?
Is this your cat?
No, I might have that on my test check, but I wanna show you these first. Alright, so this first video is just a very short, tiny clip. There's no audio. I have my speakers muted anyway. But this is Fasciola hepatica, the one that I said can go into the biceps and the liver. Hepatica is what I remember. This is what this giant tube looks like. Notice the okay. All right. So this is kind of low res for this high magnification, but this is the fluid, Fasciola hepatica. And we're looking at the bile ducts, so it's a pretty good sized thing. It could be causing obstruction, especially if it's a heavy burden like that. But you can see those suckers, and you can see the way it's kind of just manhandling the bile ducts, causing mechanical irritation of those tissues. Oh, by the way, if you guys want a podcast recommendation, TWIP, speaking of parasitology, amazing. This guy, Dixon DeConnier, love him.
Also, it's SpongeBob.
I was thinking about SpongeBob earlier. SpongeBob is a sponge, right? It's not.
Well, he was the same. Under the sea.
Yellow.
He's yellow. I did think about SpongeBob as fast, but I was like, this is something we're going to be watching after a break, so it's a little bit out of sequence, but looking at another colonoscopy, and there's a description in your notes of this patient. She's a 60-year-old woman who was presented with some mild, like not terribly severe symptoms. Let's watch that again. Okay, so they did a colonoscopy probably because of her age, and they found a couple of these giant worms, Scarus lumbricoides. So we'll be talking about that when we come back from break. This is a light infection. This is just a worm. I'll show you a heavy infection. These are very big worms. They get up to 35 centimeters. Take out the intestines and squeeze the worms out. This is not an animal. This is a human being. are underdeveloped, so they get these incredibly heavy work burdens. They get all the attendant symptoms with discomfort and abdominal issues, but they also have obstructive, like intestinal obstruction too. As you can imagine, nothing is really getting through intestines without packed worms. Okay, and then lastly, I have one more video we need to show you. This is one that we'll talk about. It's another nematode. It's the Scarasolomachrones. That's the first nematode or second we're talking about. The third one, I think, is this guy. This is Trichuris purpura, and this is the whip one. So it's a teeny tiny little worm, but you get some heavy infections. You'll see why the pathology is so bad. Whipworm infections. Also, before I press start on this, what's cool about this case is noticing the right here at like a tracker on where the probe is going through the colon, so you can track where the scope is as we move through this tour of the lower intestines, large intestines. Oh, and I just lost my internet. All right, well, I won't keep you. I'm gonna check my internet. When we come back, if I have internet, we'll watch a little colonoscopy video. I'll give you something to look forward to.
Everybody takes notes. We have a quick potential change, so I need to get everyone's input on it. Captain Wayne reached out to me and said that he only needs 3 lectures left out of 4, so he said he can cancel pretty much 4 lectures. He offered today or any of the next week. Next week we have 3 of them. One Monday afternoon, one Thursday morning, and one Friday right after the test. I was just gonna tell him to cancel the one this afternoon unless you all have different input. Does anyone rather cancel one next one? Besides, no, next week we don't have any fucking classes. Back to 2 new days.